Hello. So I'm back, and that last one was really fun. 10 out of 10, recommend. I feel like this is really helping me. It's also kind of giving me a confidence boost because I'm realizing that I understand more than I thought I did. And it's just good to recall sometimes. See, had I actually started studying at the beginning of this week like I planned to, this would have been an excellent, surefire way to make sure that I did well on all of my exams. But life happens, my dude. Okay, so I am still a little tired. You might have to give me like a few seconds, and my head hurts. I brought a cookie. Hope that's gonna be real good. That was so helpful. Like for real, for real. Okay, now we're moving into the second lecture. So let me see. The first thing my notes talk about is the linear structure of nucleotide residues in one strand of DNA. Okay, I think it's going to go into the double helix. So it says the linear structure, that's the DNA as it goes this way. Oh dear, why am I cold? Okay, the linear structure, which is the DNA as it goes this way, is maintained by covalent bonds. If I could just look this up right now. Okay. So we have one strand that's connected through Okay, perfect. So the linear structure of DNA, I'm sorry, I just had to look this up because I didn't know it. I need to find a helpful picture. You guys cannot see anything. Can you guys hear me? Okay. I hope so. So the linear structure is going to have a purine or pyrimidine base. And then it's going to have a sugar and a phosphate. That is that whole compound with the base, the sugar, and the phosphate is a nucleotide. Nucleotides make up nucleic acids. Those nucleotides have covalent interactions between the base, the sugar, the phosphate. The phosphate has a covalent interaction between that nucleotide and the sugar of another nucleotide. That sugar has a covalent interaction, a covalent bond between that sugar and another phosphate. So that is how the DNA, like one strand going this way, is bound covalently. Though when we get into double helical strands, right, then we have the bases in the middle, and they are stabilized through hydrogen bonding. So that's when the oxygen of one group, the oxygen, the nitrogen, or the fluorine, and an OH and NH, whatever, or another group are bound to each other and they form a hydrogen bond. Those stabilize the middle of the structures and they kind of hold the DNA zipped together. And then when you get into the helical transformation, there's also stabilization, I believe, going. No, I think that might be it. I don't know if there's stabilization going downwards, but we'll figure out together. Okay, so one strand of DNA is maintained by covalent bonds. We just saw that between the sugars and the phosphates. And then a double helix of DNA is stabilized by non-covalent interactions. We just talked about that. That's the hydrogen bonding that happens between the bases. Between different parts of the molecule. Okay. Proteins are made up of amino acids, which are linked by peptide bonds. We know that. The peptide bond is the N terminus bound. No, not the N terminus. We're just going to say, okay, we'll say this. A peptide bond is the C terminus bound covalently to the N terminus of another amino acid. But it's important to recognize that we start from the N terminus when we synthesize things. So it goes N terminus starting over here. Then the C terminus is over here. The C terminus connects to another N terminus of another molecule. And proteins are also folded into a specific conformation stabilized by non-covalent interactions. So normally they have I'm so tired, but I'm afraid if I don't do this now, I never will. And I don't want to flunk out of school. And I definitely don't want to study for a neuro exam 2 days before the exam. Okay. But basically, I can hear my heartbeat. I don't know what I was talking about. Okay, proteins. When they fold, they put the hydrophobic parts inside near each other so they can support each other through van der Waals forces, and they put the hydrophilic parts on the outside so it can be in contact with the water. We also talked about last time how this increases the entropy of the surrounding water by not having to expose it to the hydrophobic parts that would cause the water to form little like bubbles around the hydrophobic parts. But now it doesn't have to do that. It can just flow freely because it's only in contact with the hydrophilic parts. Okay. And then non-covalent bonds, these forces, are much weaker than covalent bonds. But this is a good thing because it allows them to constantly be broken down and reformed when it comes to biological reactions. And we need to do that rather quickly with less energy. So this weakness is what makes them important because it allows them to be continuously broken down and reformed and to rapidly exchange molecular partners. Easily broken individual bonds give rise to structures that have dynamic flexibility. So they're able to do more things and different things, which is necessary to their functions. So we just talked about how proteins are stabilized by hydrophobic. Actually, I'm trying to think. Actually, the non-covalent interactions in proteins. My zipper is like moving and I can hear it. The non-covalent the non-covalent interactions in proteins, I guess I can't say do more than just stabilize them, but they definitely help in DNA as well. So what they're saying is good about that is that it allows proteins to have dynamic flexibility as well as DNA. If DNA needs to be unzipped so it can be transcribed, then it can do that without having to expend a lot of like unnecessary energy. thus dynamic flexibility. That's why hydrogen bonds are good for us. Okay, so then we go into electrostatic. Electrostatic means it's depending on forces that electrical charges exert on one another. This is basically just talking about types of interaction. So there's charge, that's like assault. And then there's charge dipole. I don't have an example of that. And then there's dipole interactions. And then there's charge-induced dipole, where the charge induces a dipole or brings out a partial charge on a non-dipole having molecule. And then there's dipole-induced dipole, where one dipole brings out a partial charge on another molecule that would not have a permanent dipole. And then there are dispersion forces, which my lovely chemistry teacher basically just described to me as like, if you're standing next to someone, like you don't have to be touching them, but you know you're standing next to someone. So like that force that kind of tells you that something else is around. And then hydrogen bonds, of course, which are the strongest non-covalent force. And we will see that they're in between. Their strength is in between that of a covalent bond and those and non-covalent bonds. Normal bonds, normal non-covalent bonds, two molecules can never get closer than their van der Waals radii allows. However, hydrogen bonds are able to get closer than the van der Waals radius allows, which is not typical of non-covalent interactions. Okay. It just says that a fluid environment, and we know that water is the biggest component in cells, followed by proteins, and then polysaccharides, and then, proteins, nucleic acids, then polysaccharides. Let's check that out. Proteins, nucleic acids, then polysaccharides, then lipids. Okay, so this water allows for just molecular mobility. So it allows for the molecules to move around more. Yep. Okay, so this section kind of talks about the importance of water because it's the number one component, and it makes up a large majority of us. I think people say that we're like 70% water. So water molecules are not linear. They have a permanent dipole, a permanent bent shape to them. Water is unique because when in its ice form, it can form 4 hydrogen bonds to other water atoms. So it forms what's called an open lattice, I believe, versus in its water form, it forms an average of 3.6 hydrogen bonds. It's 3.6 because it's never like just 3 or 4, like they're always moving around, forming new bonds to each other. This gives water an interesting component where it is actually denser in its liquid form. It's actually more dense in its liquid form than its solid form. The solid form forms 4 hydrogen bonds and is an open lattice. The liquid form forms 3.4. These hydrogen bonds also account for the fact that water has a much higher boiling point than other molecules around its size, like methane, and a higher boiling point, lower melting point. Okay. Water also works well with charged molecules. Remember that the oxygen kind of has this like net negative charge. The hydrogens kind of have this net positive charge, which give rise to the dipole. So positive atoms will be attracted more towards that oxygen, and atoms will be attracted more towards those hydrogen sides. And this results in favorable interaction between charged species. I want to go to bed so bad. I'm just scared because as soon as I go to bed, I can't help the fact that when I wake up, it's tomorrow. And tomorrow is Saturday, and then there's only Sunday, and then I'm taking exams. Van der Waals envelope just basically describes the envelope around the whole molecule. It's like a dispersion force around the whole molecule. So it combines the van der Waals radii for that hurt. That was unpleasant. It involves the van der Waals radii for like every atom in that thing and the molecule. Okay. It's an effective surface area of a molecule obtained by making a sphere with the van der Waals radius. Okay. It is rare for two atoms from two different molecules to come closer together than some of their van der Waals radii. We already talked about that. We already talked about how hydrogen bonding kind of breaks that. So hydrogen bonding, the distance between hydrogen bound atoms changes or varies. But they do still get closer to their van der Waals. They get closer than their van der Waals radii allow or would say that they should be for non-covalent interactions. But they're still further from what they would be as covalent interactions, which is why it's kind of in the middle. Hydrogen bonding is the strongest type of non-covalent interaction. So water contains 3 atoms, but can hydrogen bond to 4 water molecules, like in ice. And liquid, they're randomly oriented to increase entropy. This makes hydrogen bonding networks rapidly changing. This is what we already talked about. There's a 3.4 average of hydrogen bonds in water. We already talked about liquid is denser than ice because of open lattice and has a high melting point and boiling point. Okay, so it's higher melting point and high boiling point. We talked about that too. So an H bond donor, for instance, could be an alcohol group or maybe a I don't know what they're called, but an SH group, maybe. So that's just hydrogen that is bound to an electronegative atom. Yep. And an H-bond acceptor could be something like carbonyl or any other electronegative atom, oxygen, nitrogen, fluorine, that has lone pairs. So this just gives a random fact that complementary DNA bases, so we just talked about those, formed by covalent interactions via the sugars and phosphates on the side and hydrogen bonds in the middle. So the bases form 2 to 3 hydrogen bonds with the base that they normally bind to. Okay. And then the hydrophobic effect. Okay, this is what I was talking about. So this kind of is responsible for the way that proteins form, and this helps to increase this helps to make the overall delta G of the reaction more negative, even though it's entropically disfavorable. The hydrophobic effect says that the entropy of water is increasing because you're putting those hydrophobic parts in the core and hydrophilic parts on the outside where they're contacting the water. So this effect causes nonpolar groups to come together in aqueous solutions. We can see this when we put water and oil together and like shake it. Those nonpolar groups form like little circles and they like try and group together because they do not want to touch the water. They're like, get as far away from me as possible. I don't like you. The water is also like, well, no one said I liked you either. So the people who don't like, like group A is like, we don't like them. You can't sit with us. Group B is like, we don't like you. You can't sit with us. So they're both trying to separate from each other as fast as they can. Hydrophobic effect. Hydrophobic effect is saying, hey, we know you guys don't like each other. So we're going to make it so you're not touching each other. We're going to make it so you're always separated, or we're going to try to. Amphiphilic and amphipathic. I don't know what. Oh, okay. I think they're the same thing. No, philic is loving. Pathic. Okay, I think they're the same thing. It just is like a lipid where you can have like a water-loving head and a water-hating tail. So a hydrophilic head and a hydrophobic tail, or both polar and non-polar segments. Okay. So water has a tendency to ionize. So you can go from H2O to H3O plus and OH. Commonly H3O plus is, sorry, my back hurts. Commonly H3O plus is denoted as H plus, but it's actually a water and an extra hydrogen. This happens because of hydrogen bonds, which create really close interactions. So I didn't know that. But for instance, it has a tendency to, now we know it has a tendency to dissociate. So to go from H2O plus, split apart, so that H plus joins onto another water molecule, and the OH would go hydrogen bond with something else. So it has the tendency to dissociate because it forms really close together hydrogen bonding interactions. The Kw, which is the dissociation rate of water, is 1 times 10 to the negative 14th. The Kw for OH, the base, is 1 times 10 to the negative 7. The Kw for H plus, the acid, is 1 times 10 to the negative 7. Put them together, you get 1 times 10 to the negative 14. What if I take like a 30-minute nap? You think that'll help? I just feel so, like, I feel like I've ran into a wall, but I'm so scared to stop because if I stop and I wake up tomorrow, I only have 2 days to prepare for a test in both biochem, neuro, psych, and physics. Guys, this might have to be it. I'm understanding this information, but it's important to me that I actually understand it well, or it might have to stop here and then I'll just go through a neuro summary. We're actually pretty close to done with this. I just want to make sure that I'm understanding it, and my body is like, no, stop talking to me. I'm tired of you. Shut up. I can't even go. I'm so tired. I'm gonna have to end this one after this and move on to neuro because my body might just be done with biochem, if that makes sense. So 1 liter of pure water is 55.5 moles. So in 1 liter of pure water, there are 55.5 moles there at any time. 1 times 10 to the negative 7 moles of H3O plus, 1 times 10 to the negative 7th moles of OH. So that's how they dissociate. The ion product of water is 1 times 10 to the negative 14th. That's those molecules multiplied together. Okay. And then we get into this fun phenomenon where the pH equals well, the pH equals the pKa plus the log of base over acid, but it also equals the negative log of 1 times the negative log of 1 over the acid. We don't really, I haven't encountered that reaction often, that equation often. So personally, I'm a little like, let me take it down. So let's do pH. Normally I don't fall asleep at this time, which is why I don't understand why I'm so tired. And I'm scared. More than tired. I'm scared. I don't feel like I'm cramming because I'm not like trying to last minute memorize. I'm trying to actually understand everything, but I'm running out of time, baby girl. Okay. We're done with this section now. So the dissociation constant equals the dissociation constant. This isn't even in relevancy to water anymore. I don't know what I'm talking about now. The dissociation constant equals the equilibrium of the acid, which is the acid form times the base divided by the acid. Isn't that just the base? I don't know what that equation means. So it's deriving the equation I told you. The pH equals the negative log of H plus. You saw me just write down pH equals negative log of 10 to the 1 over H plus. And now I'm writing that it equals the negative log of H plus. And now we're getting that the pH, I might have messed up. So the pH equals the pKa, which is the negative log of Ka, plus the log of base times acid. That's what I just said, and that's what we're going with. I don't know what this other foo-foo ha-ha is about. What can I do with these? Can I pin this? Perfect. It's pinned. Okay. When the amount of base equals the amount of acid, I don't know what just happened. When the amount of base equals the amount of acid, so when they have the same concentration, then the pH equals the pKa, because when we go back to our equation of pH equaling the pKa plus the log of base over acid, if they're the same number, that's going to be the log of 1. The log of 1 is 0, so you're going to get pH equals pKa. That makes sense. When the base is 10 times the acid, then you're going to get 10 over 1, and you're going to get the pH equals the pKa minus 1, plus 1, plus 1. Log of 10 is plus 1. Log of 1 divided by 10. Well, 1 divided by 10 or 1 divided by 100. Log of 1 divided by 100, I believe, is minus 1. Nope, log of 1 divided by 10 is minus 1. Okay. If the acid is 10 times the base, you get pKa minus 1. If the base is 10 times the acid, you get pKa plus 1. Okay. When But oh, I don't like buffers. Um, Buffers equal a mixture of acid and base. They're supposed to neutralize things. For instance, if you're studying a specific if you're studying a specific reaction and over time it is making acid, the buffer will be a base, so it can neutralize the product so the pH of your thing is not changing. That's more than I thought I knew, if I got that correct. So a buffer is a mixture of acid and a conjugate base that serves to maintain the pH of a weak acid or base. So serves as a source or sink for any acid or base made. So when we look at buffers, we actually get this something called like a titration curve, right? And if I can draw this out for you guys, I'm gonna think I'm a genius. So it looks like that, right? This would be our titration curve, right? And here in the middle, now this is what we don't understand. Don't ask me why. Here in the middle would be the pH of the buffer. Want to know why I know this? Because I got this question wrong in class. Embarrassment, man. It'll do something to you. So let's go to the interwebs real quick and look up buffers plus titration curves. I would do a problem, but that sounds horrendous. Gen Chem was not my favorite. Did I not get that correct? The point I just drew for you guys. No, I was right. But there's another point. My curve is off. I'll draw it in a different color so you can understand the part that I messed up. There's this other point here. Oh, nice blendy pins. And this point here is called the equivalence point. Good thing we're looking. Good looking, chicken bucking. The P. It says. Oh, okay. So this point is the PKA. That's not a K. Will you understand that, Alayna? PKA. Okay. PKA. Perfect. And that is when base equals 1/2 the concentration of acid. And then our equivalent, okay, that makes sense. Don't ask me why. I'm going to put this point in. Okay. So, yes, that makes sense. The buffer and the solution. The buffer and the solution have different equivalence points. So the buffer has its own it has its own point at which it's equal. And that is when the base is equal to half the concentration of the acid. But the whole solution also has an as a point of being equal, where the base is equal to the acid, the concentrations of both. And then there's an after equivalence point. So perfect. That point that I showed you, where the curve first initially kind of flattens out, that is the buffer's pKa. And then where it kind of forms that steep line that I'm about to show you is the equivalent point of the whole solution, where the base equals the acid. They say that that is the PI. How interesting. How interesting. Look at this. Learning new stuff, learning new stuff. Google. Yeah. All right. I want to scream. 1 second. Please hold. I need to put some events on my calendar. What's it say? Hopefully says 1:30 to 3 p.m. Delete. Yes. 1:30 to 3 p.m. IT Biochem review. And then on Monday from 8 to 9 is physics exam 1 review. Who do I have a meeting with? Oh no. No. Okay, we're skipping this week. We're just gonna move. This is my father's birthday. See, it will be a very busy week tomorrow. I mean, because I have tests and then I have to catch up on all of my work. I have a lab report to do. Finish your lab report. Sorry, I just realized I have lots to do. Oh, that makes so much sense. Sorry, give me a second. I was like, why is everything off? What is happening here? I am not going to move this one. Okay, perfect. Okay. I still haven't done that. Okay. So this is a titration curve. Originally I showed you guys that one in bluish green. Yellow, the orange is everything that I missed. So this is the pKa of the buffer, where that curve first originally flattens out. And then over here is the equivalence point, where the pKa of the acid is equal to the pKa of the base. And then up here we have the pKa of the acid that we're buffering. The pKa of the base is when the base is equal to 1/2 of the acid. Okay, and that finishes up lecture 2. Let's see what lecture 3 is about. Because if I can do 3 and 4 real quick, I'll come back on for another section tomorrow. So I'm going to actually finish this first section on this. We're going to move into lecture 3. So we just finished lecture 2. We're looking good. We're cooking chicken. Good looking chicken. You know what I'm saying? We're doing well, and I am well pleased. And then after this, I'm going to look through some bio stuff. Do a psych midterm, do a biochem exam practice. Please. And then maybe answer neural questions. That might be highly pushing up. My dad's birthday is on Wednesday. I know he's going to want to see me, as he should. But I'm going to be so busy that week. Balancing a personal life with school is an actual nightmare. Let's finish half of the material, and I'll come back and do the other half tomorrow. I still need to do a neuro one of these. We're not doing that tonight. Okay. So this chapter is about amino acids. We love them. Okay. So there are 22 naturally occurring amino acids. I would draw them all out for you, but I don't have time. Glycine side chain has 2 hydrogens. It is the only amino acid that's not a chiral center, that doesn't have 1 chiral center. Alanine has a CH3 group as its one, and then we go into valine. Valine has a CH group with 2 CH3 groups coming out of it. Leucine has a CH group that goes into a CH. I'm trying to count my hydrogens. Leucine has a CH2 group that goes into a CH group that goes into 2 CH3 groups. Isoleucine has a CH2, has a CH3, CH2, CH3. I literally cannot go all through all of them with you, but I'm pretty sure, 97.8% sure I know all of them. So let's do this. Okay, the more hydrophobic amino acids, such as that whole row I just told you guys, that would be the glycine, alanine, valine, proline, leucine, isoleucine groups. Also, phenylalanine and maybe tyrosine because it has too many carbons, and maybe tryptophan, I believe. Let's check this. So, yes. Okay. So I missed some. I missed 3. No, I missed 2. Nope, I missed 1. Methionine is also methionine does not have a methionine has a sulfur like cysteine. Ew, but is not, oh gosh, that was making my tongue itch. Oh, why is my tongue itching? But is not a chiral center like threonine or isoleucine. Okay, we're good. Those are hydrophobic. They're usually found within the core of folded proteins. We already covered that. So they're shielded for water. We covered that. So there are terns. Terns are sites. Wait, are we in proteins? Hello, can we skip protein structures? Whatever. So if you have alpha helix here, there will be a tern and then another alpha helix. Fun fact, proline cannot be in an alpha helix because of its weird structure. Has a weird structure where the N terminal is attached to the side chain, results in like it not being flexible. Like I am a proline, I am not flexible. So it breaks alpha helices. It's also not great in beta sheets. So you probably won't find them in alpha helices or beta sheets, but you will find them in turn because their awful inflexibility, I don't know if that's a word, makes them bad for makes them good for turning. I think as well as glycine's flexibility. Okay, so terns are site in a protein structure where the protein polymer folds back on itself. Psych, I was still talking about terns, but I was not talking about the right type of terns. It's talking about like take a piece of paper, crumble it. Terns. Okay. Glycine can form tight glycine can form tight terns. So it's frequently on the surface, and so can proline. So those are kind of our exceptions. So we just talked about those hydrophobic. We just talked about how hydrophobic amino acids are normally in the center, but glycine and proline are great for turning. So those two are normally on the outside. Okay. Phenylalanine, valine, leucine, and isoleucine are the most hydrophobic amino acids. Phenylalanine, leucine, tyrosine, and tryptophan can also be cut by cyanometrypsin. Okay, different things. So those are the most hydrophobic amino acids. I did not know that, nor do I know how to memorize that. We can add that to my sheet. Maybe it'll help you. This sheet is not for clean. This sheet is not for cheating, pero it is helpful for just like, adding information. So phenylalanine is the most nonpolar. So is leucine and isoleucine. And valine. Okay, so that's what that looks like, if you guys were wondering. She's pretty. I made her before the quarter even started. All right, so those ones are the most hydrophobic. The aromatic amino acids, so those are the Wow. Those are tyrosine, tryptophan, phenylalanine. Those are the ones they mean, but there are other ones with rings. Histidine has a ring. And that's it. That's correct. But they're not aromatic. Histidine may be aromatic. I think histidine is aromatic, but it's not what it means. So the aromatic amino acids absorb light in the UV region. This is fun. We see that tryptophan and tyrosine absorb light around 280 nanometers, whereas phenylalanine is around 230, I believe, 250. Phenylalanine is around 250. So if we're looking for a UV absorption that's around 280, we need to be looking for tryptophan or tyrosine. But if we're looking for one that's just above 250, we can look for phenylalanine, tyrosine, or tryptophan. Those are the ones that have UV, they absorb UV light, and we can see them. So polar amino acids are everything else. Let's see if I can name them. So headache. Polar amino acids include histidine, lysine, arginine, asparagine, glutamine, glutamic acid, aspartic acid, also threonine, cysteine, serine. I believe that's it. Yup. Good looking. Okay. Polar amino acids like serine, cysteine, threonine, asparagine, and glutamate or whatever can form H bonds and are found on the surface. So those ones are hydrophilic. They can come in contact with the water, so they're found on the surface. Fun fact, when we talk about protein folding in an alpha helix, we see that it's folded in such a way where the first one, where every 3 to 4 amino acids should be non-polar. And this would have them facing the inside of the structure versus the outside. I did not explain that well, but yeah. Okay, so we basically have said that non-polar atoms are in the inside of protein structures where they don't have to contact water. Polar ones are on the outside. Exceptions, we have proline and glycine, which are amazing at turns and get to be on the outside. The aromatic ones can absorb light. All of them can absorb light, but tyrosine and tryptophan can absorb more light than phenylalanine. Histidine side chain, histidine and cysteine side chains are very close to physiological pH. So histidine is at 6, cysteine is at 8.3. This makes them very helpful in reactions that require like ionizations, I think, and like under physiological pH. But let's read into that. So histidine side chain has a pKa near physiological pH. So it's often involved in proton transferring during enzymatic catalysis. We actually, yeah, she is, because histidine is a general acid / general base when it comes to acid-base catalysis. Would you look at that? I'm scared to read about Tim Burrells. No, that's tomorrow. Tomorrow's going to be a hard day for you guys. go. Your girl has a headache. General acid and general base catalysis. Yep. Perfect. Histine's perfect for that because it has a pK near physiological pH, and so does cysteine. Okay. Basic amino acids, those are glutamic acid and aspartic acid, are strongly polar and are found on the exterior where they can be hydrated by the surrounding aqueous solutions or in substrate binding clefts of enzymes where they can interact with polar groups on the surface that bind to the enzyme. Okay. We also see this in the oxyanion hole, kind of, but those are just having NH3s all pointed directly in, which is giving a positive charge so that the oxygen bounds to it. Let's just make sure I'm getting basic amino acids correctly. Basic one should be, oh, yep, I was wrong. Okay, so I don't know why I just responded to basic amino acids with aspartic acid and glutamic acid. Basic amino acids are histidine, arginine, and lysine. Okay. They're strongly polar and they can be found in enzyme active sites or on the surface because they love to contact water. There are there are 20 amino acids, but there are two special ones, selenocysteine and pyrrolysine. That's all I have on them. All prokaryotic proteins start with the N. So this is a backstory. So most prokaryotic proteins, notice that we are eukaryotes. So us and plant cells, animal cells and plant cells are eukaryotes. Prokaryotes, most of their proteins start with N-formyl methionine. Methionine is not a chiral center. And it's nonpolar. Okay. Most eukaryotic proteins, that's us, we start with methionine. So most prokaryotic ones start with N-methyl, start with N-formyl methionine, and most most prokaryotes start with N-formyl methionine. Most eukaryotes start with methionine. That was a very good point that I did not know. I like how we almost have covered the first 5 chapters. I'm saying that as we're in lecture 3. And this is the difference. You see, the first half of the quarter always easier than the second half. Okay. Amino acids are building blocks of proteins. Proteins are one of the most important and abundant biomolecules. Going back over the order, we have water, proteins, polysaccharides, lipids, other stuff. Okay. Proteins are biopolymers. Going back over biopolymers, we have everything but lipids. Lipids are not biopolymers. They're not made up of monomeric units that are covalently bound to each other. Okay. So, then we have the basic structure of an amino acid. All amino acids are L. L. Yep. All are L. Most are L. We love L. We don't like D. Okay. So here we have the basic structure of an amino acid, where the sides of the amino acid are coming out and the back is going in. Question mark. Let me check this. I don't always understand the shortest verification. That's not the right word. Oh, okay. The information I'm looking for was this one such as it just wants to double check. Okay. So the side groups, where are you? The side groups. So these two groups, the C terminus and the N terminus are coming out versus the side chain and this extra carbon are going in. And that is for all, that's the structure that we draw proteins with. The Zweiter ion is the ion in which the is shown when the protein is at is pi, which is the concentration where the net charge of the protein is 0. Before it's pi, the protein will have a positive charge from the N terminus, at least one positive charge. Of course, if we're going to the bases, they have a more positive charge. After it's pi, it will have a negative charge. At least one. Going into the acids, they can have more than one negative charge. Okay, so cysteine has a sulfur on it. Remember, the pH of that one is 8.3, close to physiological pH, and that is a good nucleophile. So it's good at coordinating metals and can form disulfide bridges. Ouch. Okay, that is good. Let me just check some information here. Where is my ion complex? It's not there. Ow. No, okay. Okay, so cysteine is good at coordinating metals because it has a nucleophilic SH. It can also form a disulfide bond, and that is called cysteine. Cysteine is cysteine that's connected to itself via a disulfide bond. Histidine is often involved in acid-base catalysis. We already talked about this. The side chain is also good at coordinating metals. That is a great thing for us. What do we have in myoglobin? Yes, histidine is responsible for. Okay, so in myoglobin, there are 4. There's a pyrrhus ring. Well, it's the heme is coordinated by 4 nitrogen atoms, 1 histidine, and if it's bound, 1 oxygen. So here's an example that we see of histidine coordinating a metal. which is the Fe 2 plus. Yes, ma'am. The other one has 2 carbons and something else. Okay. Aspartic acid and glutamic acid, we just talked about those as bases which love to be on the outside where they can be hydrolyzed or in enzyme active sites. Fun fact, when we get to serine proteases, we will see that that's not true, is it? That's not true. Please ignore what I just said. Okay. So aspartic acid and glutamate or glutamic acid, their side chain carboxylates occur often in active sites of enzymes. We already talked about this. And they coordinate things like calcium and magnesium. So those are just like positively charged ions. Aromatic amino acids, as we have said before, absorb UV light. And they're used to measure, they can be used to measure protein concentrations. I did not know that. Did I know that? I don't think I knew that. Protein. No. Protein. Not there. Protein. There we go. Do we have anything about UV? Okay. Oh, to measure the concentrations. So this is the end step. Not that far in the end. Ouch. These right there. Methods for determining protein concentration. UV spectroscopy based on UV I can't say it. It's not going to work. Absorption of aromatic side chains, tryptophan and I wrote tryptophan and tryptophan. Tryptophan and tyrosine are at 280 versus this one says phenylalanine is at 220, but I believe it's actually 250. So this shifts the absorption of proteins upon binding to specific method. Okay, so yeah, it can be used to determine protein concentration. So keep in mind that the aromatic amino acids can be used to determine protein concentration because they have UV signals. And remember that aspartic acid and glutamate are found in active sites, and that histidine helps coordinate metals and can act as a general acid or base in general acid base catalysis, and that cysteine is also good for coordinating metals. And I don't remember what else cis is good for, and can also form disulfide bridges. Okay. All right. Okay. Amino acid PKs are an average of the values change are amino acid PKs are averages. The values change depending on where they are in a protein. Okay, that makes sense. Now let's try and think through this. If an amino acid is next to more acidic proteins, the pKa is going to go up so that there's attraction versus repulsion, versus if it's closer to more basic proteins, the pKa, oh crap, what did I just do? If it's closer to more acidic proteins, the pKa will go up so that it is more basic. If it's next to more basic proteins, the pKa will go down so it's more acidic. That's what I'm thinking. Now let's read this. Base will raise pKa. What did I just say? Base will raise pKa, so there isn't a charge repulsion in negative environments. Okay, all right. I was thinking about it the wrong way. Okay, so we're only talking about acids and bases, for example, here. So let's say the protein is in a negatively charged environment. If it's in a negatively charged environment, their pKa, oh, why does it say raise? If it's in a negatively charged environment, their pKa will go down to become more positively charged, so there's no repulsion. And it will go up in more positive environments. My notes say the opposite, but I'm pretty sure I'm understanding it the correct way. Let me ask. It's bright. It's very bright at this time of night. So I think tomorrow. I really don't like sending people my notes because, fun fact, I can't write or spell anything for the life of me. See, I already spelled average wrong. Let me do this. Nope, let me do this. Go. I have this section of my notes that say that the amino acids PKs are an average and that they change in response to the environment, which makes sense. But my notes say that a base will raise its pK, and I'm understanding raise its pK to mean become more basic. But it's saying a base will raise pK, so there isn't a charge repulsion in a negative environment. But if the environment is negative, shouldn't the base lower its pKa so it becomes more acidic? So that there's a charge attraction. Does that make any sense to you? What do you think about this? Okay, perfect. That's done. Um. Okay, all amino acids except glycine have a chiral center and form D or L enantiomers, but L is most common. We did already say that. We are on top of our stuff. I am getting off of here at 12 a.m., which is 30 minutes away. So L is L is different. That was my head hurting. Oh, look at the nails. Look at those nails. Okay. L has look at the tattoo. Look at the tattoo. Anyways, L has the N terminal and the H of the carbon that's connected to the side chain coming out. But the C terminus and the side chain are going into the plane. Oh, let's remember that. Let's also remember that in normal amino acids, I'm going to say proline and glycine are the exception here. Glycine because it doesn't have a chiral center, and proline because its chiral center, it has a chiral center, but the side chain is connected to the backbone. Normally, when we draw it, the H is going to, I can't really like put my hands up and show you guys because it might revert the image. So I'm going to do it on my direction. Just know that this is my left and this is my right. So normally we see the dashed hydrogen on the left and the wedged side chain on the right. When you flip that upside down, because when you draw a protein, the amino acids kind of like one side chains up, one side chains down, one side. When you flip it upside down, the dash hydrogen is on a wedge on the left, and the wedged side chain is on a dash on the right still. Just letting you guys know that. Okay, so if there's amino acid and it's not proline and it's flipped downwards, as in the side chain is facing downwards and the side chain is wedged, it is L and it is not D. I know that also because I got that question wrong, but I got that wrong because I wasn't paying attention. Okay, let's go back over this. Amino acid derivatives and other chemical modifications. Neurotransmitting amino acid derivatives. Oh, yes, this is important. Okay, so there are amino acids that are the derivative. No, there are neurotransmitters. There are neurotransmitters which are derived from amino acids. We have GABA, histamine. Histamine's easy. What's histamine derived from, guys? Histidine. And basically, we have GABA, histamine, and dopamine. Dopamine is derived from tyrosine. There's a form named L-Dopa, which is this cool, well, I don't know about cool, but it can pass the blood-brain barrier, whereas dopamine can't. So in cases like, I believe, Parkinson, where you're not getting enough dopamine and then your movements are off, they can inject L-Dopa into you or do something more sciencey than that. Because, yeah, no, this isn't neuro. Not yet. But, okay, so GABA is, let's look at it. GABA is derived from, oh, I can't even see what it's showing. Okay, GABA appears to be derived from aspartic acid. That's what I would say. Yes. Nope, from glutamic acid. GABA is derived from glutamic acid. Perfect. Okay. What's going on up here? Oh, sadness. Okay. We have covered that. And then it says there's also thyroxine, probably derivative of furan. Okay. Oof. Oh. We're gonna stop at 4. That's what I said, right? 4 today, 4 tomorrow, 2 the last day. So we're doing. I don't think I can do 5. My head is literally killing me. Dude, I'm not be able to do 4. 4 is, shoot. We might stop at 3. I have a headache. Okay. Amino acid side chain modifications. So there's phosphorylation where a phosphorus group is added. There's carboxylation where a carbonyl is added. Hydroxylation where an OH is added. For instance, we saw that histamine. No, dopamine was hydroxylated. Nope. There is a portion of tyrosine that was hydroxylated in the derivative of dopamine. That's how I would say it. Yes. And then methylation adding a CH3 group, and then acetylation adding an acetyl group. So, O-phosphoserine. Sorry. Probably ortho. I don't know. Is important in cellular signaling. I think that was just an example. Frequently used to turn proteins on and off. Oh, that's the thing. Phosphorylation is also like commonly used to activate proteins or turn them on and off. So that's believed to occur in 30% of proteins, which doesn't sound like a lot, but you have a lot of proteins, my dude. Okay. Carboxylation is there's gamma carboxylamate, I believe, and it's less common but allows molecules to bind to calcium itself and plays an important role in blood clotting cascade. These are just examples. Carboxylation does not have to be this. And then hydroxylation, an example is 4-hydroxyproline. Fun fact, proline and hydroxyproline are both very common in collagen. Collagen is a structural thing. It's hard and it makes up things. Yeah, it says major component in protein collagen, which constitutes connective tissues in the body. And then methylation, there's 3-methylhistidine, which plays a role in muscle cells, and then acetylation, and it's important for gene regulation. So those are the different ways to modify amino acid chains. Majority of them are reversible. Yes, reversible modifications of specific amino acids is incredibly important for signal transduction. That's where the knowledge stops. I don't know. I know it can turn it on and off and stuff like that, and that's why it's good. Protein kinases, they cut proteins. And protein phosphates cut the phosphorus group off of protein. So they remove the phosphorus groups. So they are able to reverse phosphorylation. Protein kinases attach the phosphor. Oh, I was wrong. Okay. Protein kinases attach phosphoryl groups to specific. Alrighty. Protein kinases are able to attach a phosphoryl group onto molecules that have a hydroxyl group. So they replace the hydroxyl group with the phosphoryl group. So we see this specifically in serine, threonine, and tyrosine. Note, threonine is does have 2 chiral centers like isoleucine. And it typically attaches to the hydroxyl group. So remember, protein kinase attaches phosphoryl group to hydroxyl groups of serine, threonine, and tyrosine. And yes, protein phosphatesis removes these phosphoryl groups. After phosphorylation, at one or more sites, the properties of a protein differ. So if you keep changing things up, things aren't going to be the same. Key mechanisms allowing cells to respond to external stimuli. So, okay, the fact that the proteins can change functions or differ allows them to survive different stimuli. This has coffee in it. I want to drink it because it tastes good, but I can't stay up a second longer. My head is hurting. Probably from this amazing study sesh. Okay, protein glycosylation. Remember that we said in the first page, the first part of the review session, that glycolysis was the breaking down of sugars. And it changes them to Ethanol. So, common type of modification of side chains on proteins is protein glycolysis. Different sugars are covalently attached to residues of proteins. So, sugars can be covalently attached to residues of proteins. Here, this is so interesting because sugars can be covalently attached to sugars. This leads to the biopolymer carbohydrates, or amino acids can be covalently attached to other amino acids, which leads to the biopolymer. Uh, oh my, I'm so sorry. I'm so sorry. Which leads to the biopolymer of proteins. I'm so sorry. I know I did it before, but I just was. I don't think I was expecting that. That was kind of gross. I'm sorry. And what do we have here? So sugars can covalently attach to different red proteins, and this can cause them to form like large branch sugars. We did read before that sugars have branch connections. We are almost done, my dudes. I'm not going to do a section 4. We made it through 3 lectures out of 10. We have 7 more. Okay. So in eukaryotes, almost all secreted membrane-associated proteins are glycosylated. That means almost all of them have a sugar attached to them. Protein glycosylation. Sorry, these words are getting real complicated as my brain is like dying inside. Protein glycosylation is important for many functions. Okay. It forms the extracellular matrix in higher organisms. So that I need to remember versus just reading. So protein glycosylation is a common type of modification to protein side chains where different sugars are covalently attached to protein residues. And this is good because it helps form the extracellular matrix, which is a mixture of proteins and oligosaccharides. Oligosaccharides, as we already talked about, are saccharides. I always want to say they're proteins. They're saccharides that bind to cells and help them orient themselves. The extracellular matrix is a key property to cell position in respect to other cells, which is so interesting because it is a mixture of proteins and oligosaccharides. So, of course, it's going to help cells orient themselves. And it's crucial in very complex processes like developmental biology and wound healing. Guys, the brain is like, you're using me. It hurts. It hurts. There's O-type glycosylation and N-type glycosylation. So O-type is where an oligosaccharide is attached to a certain chain of serine or threonine. Those two both have alcohol groups. They are, one is polar and one is nonpolar, neither aromatic, and they both have alcohol groups. And O-type glycosylation is where, my eyes mush. O-type glycosylation is where an oligosaccharide, which helps it orient itself, is attached to a serine or threonine. N-type is when an oligosaccharide is attached to the side chain, the nitrogen. Okay, so in O-type, the oligosaccharide is attached to the O, and then the N-type is attached to the N of aspartate, asparagine. So it's not the same molecules. O-type happens in serine and threonine, and it's where the oligosaccharide is attached to the O. N-type glycosylation happens in asparagine, and it's when the oligosaccharide is attached to the N. My eyes feel like they're burning. In all eukaryotes, they start with enzymes attaching long chains, pentasaccharides. So that's 5 pentasaccharide cores. So, in all eukaryotes, remember also that eukaryotes, what did it say? Most proteins start with methionine. We talked about this in response to prokaryotes, which starts with informal methionine. Yep, perfect. Just saying. Okay, so glycosylation starts with a pentasaccharide that's then cut down after that. So, yeah. All right. We've made it a long way. We have one more section. I don't know if I can do it because my eyes are like burning. Um, Lecture 3, the peptide bond and biologically active peptides. Amino acids are joined together by condensation reactions releasing H2O. We talked about that in the first video. The pKa of the N terminus is around -8. What? Is around 8. And the pK of the C terminus is around C. So this is different from the actual acids themselves, which have N terminus and C terminus pKs. This is a generalization when you're talking about a whole protein chain, not a chain with the residues. Okay. Glutathione. Glutathione is a reducing agent in cells. So it actually has one bond that is not a real amide bond. It's actually it's connected to the side chain of glutamic acid instead of to the C terminus. Yes, ma'am. But still, it has a disulfide bond that is great at just reacting with oxygen because any free unreacted oxygen in our cells can be very bad. So this molecule works to react with oxygen. So it's not free in our cells to react with other things, kill us. So it plays an important role in maintaining the reducing environment in cells. Ribosomes are the homes of proteins. There's lots of them in prokaryotes. They were purple in the picture my instructor showed me. Okay, but ribosomes consist of 30. Ribosomes consist of 66% RNA and 34% proteins. And then it has how much they weigh on here. I don't know if I need to know that. We good? 66% RNA, 34% proteins. Ribosomes, no. Ribosomes bind to what? Ribosomes bind to mRNA, which contains a codon encoding the sequence of amino acids and polypeptide chains. What the heck's happening here? Okay. Can you please take him out tomorrow? I need 1 day of sleep. Just one day of at least 8 hours. I can't ask you that. You've just been working for 17 hours. Never mind. I got him. Okay. Sequence of amino acids and polypeptide chains. mRNA. Okay. I don't know what I did here in my notes. Am I talking about ribosomes still? I think I'm talking about what happens inside ribosomes. So inside ribosomes, once again, they consist of 66% RNA and 34% protein. And then inside of them, they bind to mRNA, which contains codons encoding the sequence of amino acids and polypeptide chains. And then that mRNA binds to tRNA, which is charged with amino acids, which is changed with amino acids. And then that translates the genetic code. So it takes mRNA proteins, translates them, and converts them into building proteins. Oh, that was a lot of information that my brain can't understand because it's too late. Ribosomes, they take they translate the genetic code, and then they take mRNA and they take the mRNA takes some proteins, translates them through tRNA, and converts them, then builds more proteins. And that was the end. So tomorrow we'll kick off with the fourth lecture. Actually fun stuff, but it's 3 pages long and I couldn't even get through the last 2 lines of that last one. But I'm excited for tomorrow. We're not gonna do that first thing tomorrow. So in the morning when I wake up, we're gonna talk about this bad boy. This bad boy's name is neuroscience. There's not as much information to talk about, so I will definitely be finishing all of the information tomorrow. And then I probably won't be making another biochem vid until what is happening? What's he said? Until the nighttime. For a real one. Because let me just see what I got for you guys tomorrow. Psych, that's his light transaction. The eye stuff, I really feel like is gonna interest you guys. Maybe I am like psychopathic. Who's gonna watch this? Why am I saying you guys? But if you do, hi. Okay. We have this many pages of neuro. Fun thing about neuro, it actually is fewer pages. Yeah. This information will break your brain. Not you, anybody. Biochem's fun though. I forgot how fun biochem was. But yeah, we'll be working on neuro first thing in the morning. And we actually have a lot of neuro to get through tomorrow. So I'm probably just gonna come on tomorrow, read you guys the story, and then also work through some just basic questions applying that knowledge. And then do biochem toward the end. Thank you guys for sticking with me throughout the study sessions 1 and 2 for biochem. There will probably be 2 more for biochem tomorrow and like 3 more for neuro tomorrow. Yeah. Have a good night.